All right. This is take 2 of Journal Pub. Hi, everybody.
Hello.
We thought a lot about changing the format and all the ways we could change the format and decided not to. We're drinking Hotty Totties.
Hot toddies.
Hot Toddies.
But they can be hotty toddies. That's fine.
And they are warm.
Courtesy of Zach. Yes. It's officially November, so it's warm drink weather.
Oh, it's like the tea that you drink when you're sick but alcoholic.
Mm-hmm.
It's very good. I'm Linnea.
I'm Bez.
I'm Zach.
I'm Megan.
And I'm Linnea.
We're reading Inhibition of ATM Kinase Rescues Planarian Regeneration After Lethal Radiation. These are some squeaky chairs. We were worried about the dropping of the water. It's going to be just the chair squeak the whole time.
I'll try to stay still.
Oh, it's okay. You guys know these people. So does someone want to say the authors' names?
Yeah, so the first author is Divya Shore. You want me to go through all of them?
Yeah.
Kwanse Wang, Bhargav Sanketi, Justin Tapper, and then last author is Carolyn Adler.
Carrie Adler. We're reading this paper because I did a course with Carrie, the last author, over the summer in which we used Planaria. And then it turns out that Megan did a rotation with her, and then Zach took the bus with her once. You asked me a few minutes before we started if I had read this paper before. I'm sure that I said that I read this paper.
I think that was a large part of the reason why Bez was hesitant to do this paper for Journal Pub, was that it might become apparent that it is a first read through.
Okay.
I definitely have skimmed through this paper before, but I really felt probably over the summer that I could get away with the Sparks Notes version and just ask repeatedly to Carrie's lab what was actually going on. But I've become very interested in doing some like Planaria experiments myself. And then also I kind of feel like Journal Pub is a time where we can read papers that might not be necessarily that we could read in Journal Club. And so, yeah, because this paper is like a little bit close to home, maybe it's not super appropriate for Journal Club. I don't know. Maybe that's bullshit.
Maybe this is too basic of a question, but what is a planaria?
Zach, you said you used to raise them.
No, I had one.
You had
Planum
one.
is a worm.
Wait, and why did you have
So
one?
in school, we did an experiment in biology on the planaria. And then I must have been in January or something. And then the teacher was like, oh, you can take them home if you want. If you bring it back on the last day of school, I'll give you a bonus point.
One bonus point.
One bonus point. So I took it home and I kept it alive. I fed it mostly egg yolk. And it went from like a nice black color to very gray by the time I returned it, but it did survive. I don't know if I got my bonus point.
I think we like fed them some sort of meat mush.
I know, like when I rotated in the lab, I think they feed them calf liver or cow liver.
Yeah, that sounds right.
And then, so like every once in a while, like it might even be like every 2 years or something, they go to a butcher and they get a liver. And then they have like a day where like the whole lab like processes the liver and like blends it up and like aliquots it and puts it in the freezer. And I think they
familiar.
Yeah, we used to do a very similar thing for cow brains.
Yeah.
Really? Why?
For the tubulin prep, because the neurons are like such a rich source of tubulin.
Okay. And so you would just go to the butcher?
We just go to the butcher. We go to a kosher butcher.
Nice.
And there was very conspicuously pigs on the killing floor of that kosher butcher shop. But we did kosher butcher because normally you like this thunk in the back of the cow head, but that sometimes like splinters the skull into the brain, which ruins the tubulin prep. So we went to a place where they hung the cow up and they slit the throat.
It's very graphic.
Sorry, it was
Very tough for the like young first-year graduate student. You
Yeah, it
go
was
to the butcher at 5 in the
terrifying.
morning.
Do you have to be there like when it's
because then you have to go
happening?
fast, because you have to get the protein. Like every minute that you wait, like someone did a curve once. It's like an exponential decay of how much protein you're getting, and so like every minute you wait is like you're 10% less protein.
And
right
we tried to do it in California, and they won't let you go to the butcher, and they just deliver you the brain 6 hours later, and we got no protein.
Oh, so
Yeah.
it does. You
matter.
had to be.
I
in
don't
real
think
time.
this is that hardcore. I think they probably just order the cow liver or buy it at the store.
Yeah, and I think one gets them. The worms are pretty small, so I think one big cow liver gets them probably a year's worth of food, which is very nice.
So is there actually alcohol in the Hotty Totty? Because this is really this is what it tastes like.
Tastes like
It's
tea.
like tea.
Yeah.
Yeah,
It's
not just tea, but specifically if I'm sick tea with the lemon and the honey and the, well, we'll see. Okay.
Yeah, there's whiskey.
Okay, so we're gonna read up to.
There is not whiskey.
If not whiskey,
I forgot to put the alcohol.
I knew it. I
Wait,
knew
so
it.
we're just drinking.
Yep.
Should we pour?
Should we?
Yeah.
Should we
I'm just
use
gonna-
this opportunity to?
I do think we maybe should measure.
You think we should measure?
I would like to know how much going in.
All right. While this is happening, I'm gonna read the abstract.
Okay.
All right. As stem cells divide, they acquire mutations that can be passed on to daughter cells. To mitigate potentially deleterious outcomes, cells activate the DNA damage response, DDR, network, which governs several cellular outcomes following DNA damage, including repairing DNA or undergoing apoptosis. At the helm of the DDR are three PI3-like kinases, including ataxia telangiectasia mutated, ATM. We report here that the knockdown of ATM in Planarian flatworms enables stem cells to withstand lethal doses of radiation, which would otherwise induce cell death. In this context, stem cells circumvent apoptosis, replicate their DNA, and recover function using homologous recombination-mediated DNA repair. Despite radiation exposure, ATM knockdown animals survive long-term and regenerate new tissues. I'm really worried that I'm clipping a little bit. Am I orange? Yeah, well, we'll just have to get me on another thing. These effects occur independently of ATM's canonical downstream effector P53. Together, our results demonstrate that in Planarians, ATM promotes radiation-induced apoptosis. This acute ATM-dependent apoptosis is a key determinant of long-term animal survival. Our results suggest that inhibition of ATM in these organisms could therefore potentially favor cell survival after radiation without obvious effects on stem cell behavior. Did everyone get that completely?
Oh yeah, 100%. I wasn't at all distracted by the pouring of shots.
Right, so now there is alcohol in the tea.
I do think it tastes a little bit different now.
Yeah.
Okay, I'm going to try it again.
But I was excited to hear the word
Completely
kinase.
different.
I was excited to hear the word kinase, and I was excited to hear the protein P53.
Okay, first, PI3-like kinases, did that mean anything to anyone? Other than it's a kinase?
Yeah. Other than
PI3?
I don't
No.
know what PI3-like means now.
All right, does anyone know how to pronounce ATM?
I think you're pronouncing it correctly.
Ataxia telangiectasia
No,
mutated?
ATM.
ATM, yeah. Why are you excited about P53?
I'm pretty sure P53 forms condensates. And that, yeah, it's an interesting, it's in a lot of signaling pathways, and so it's an interesting protein to see where it,
Okay,
when
so
it condenses.
to
it makes me
recap,
think cancer.
Cancer. Yeah, I know Nikhil is interested in it, and he's MD, PhD, so that sounds
Must be cancer.
possible.
ATM is part of the DDR, the DNA damage response. If they knock down ATM, then the cells don't kill themselves after radiation.
Okay. Can I read the introduction now?
Oh, yeah, sure. I was just re-summarizing the abstract real quick for you.
Oh,
But if you don't want to know what the paper is about, just read the introduction. Let's go.
okay, I'm going to read the first paragraph. Stem cells fuel the production of new cells during homeostasis and tissue regeneration, despite continual exposure to environmental and organismal insults that increase the probability of acquiring mutations with each round of cell division. Mutations can alter the output of stem cells, often adversely, by driving senescence, premature differentiation, or promoting tumorigenesis. Whether stem cells undergo cell death or repair, their DNA depends on their cell cycle status. the intrinsic turnover rate of tissues, and the probability of entering quiescence. To combat the accumulation of DNA damage and prevent I'm never going to be able to say this word, tumorigenesis.
Isn't it tumorigenesis? It's
Tumor-
tumorigenesis.
Yeah, I understand what it is. I
Okay.
just can't say the word. Adult stem cells engage different arms of the DNA damage response, DDR, pathway, depending on the tissue where they reside. So is DDR...
Yeah, we do kind of have to come up with a
I don't actually see a lot of DDRs.
drink thing.
But I do think
ATM.
ATM is
ATM
also
is
a
probably-
candidate.
I think ATM is going to be too much. ATM is going to be in almost every sentence.
I think that's okay.
You think it's okay to have ATM in almost
Well,
every
I think
Well,
I
we
think
run out of a drink. Well,
well,
All
yeah,
right,
we'll
all right.
pause. yeah.
ATM is the drink word.
But it's not in this first paragraph, so you're okay. You can take a break.
Okay.
Depending on the tissue where they reside. For example, mouse hair follicle stem cells rely on increased expression of DNA repair proteins and anti-apoptotic factors to repair and evade apoptosis, while stem cells in the small intestine exhibit increased radiation sensitivity due to reduced expression of anti-apoptotic proteins. The difference among stem cell types raises the question of how stem cells cope with DNA damage and the underlying mechanisms that determine their fates.
Man, I feel like I was like kind of following along for the last one that we did while other people were reading. This time, I think it's because I'm panicking that we're not recording, and I'm like, "Are we recording? Oh, we're recording. Are we recording? Are we recording?" So I'm having okay. So
Having this thing on the table is too.
that paragraph said different stem cells have different DNA damage response amount, which controls whether or not they apoptose. Is that a summary?
Yeah, and I think like stem cells generally don't prefer to apoptose.
Mostly, I feel like I just learned a lot about their writing style.
I like this rule because we always talk about when you nuke a planaria, only why haven't you nuked the stem cells? And so stem cells don't want to apoptose. I like that rule. Okay. Zach, you're up.
in many species, the choice to repair DNA or initiate apoptosis is coordinated by three apical kinases: ataxia telangiectasia mutated dependent protein kinase. Oh, I skipped a line. Ataxia telangiectasia.
I say we just go with the acronyms.
And RAD3 related ATR and DNA dependent protein kinase, DNA PK, which are members of the phosphoinositide 3 kinase related kinase family, PI3K related kinase family. ATM and ATR serve as important checkpoint regulators that stall the cell cycle upon DNA damage. Human cells lacking ATM are often hypersensitive to DNA damage induced by radiation, resulting in the cellular accumulation of double-strand breaks. Cellular outcomes following DNA damage can vary depending on cell type. For example, ATM double minus minus, how should we say that?
You are
loss,
genetics people.
knockout, ATM knockout, thymocytes and postmeiotic neurons are resistant to radiation-induced apoptosis, while ATM knockout hematopoietic stem cells fail to self-renew long-term. Collectively, these results suggest that tuning apical kinase activity in different cell types could be a strategy used to manipulate stem cell outcomes.
What do they mean by apical kinase activity? So I'm really tuned in to the word apical these days, especially if combined with kinase. What is the word apical doing there?
I assume it means a kinase that is close to the cell membrane.
A kinase
No?
that is close to the cell membrane is an apical kinase. Why is it a
Maybe-
basal kinase? But, you know,
Oh, I get it. That's a good point. I don't know. That sounded.
I just thought it was a
that sound-
kinase that was on the apical side.
but why would these results suggest that tuning apical kinase activity in different cell types could be a strategy to manipulate stem cell outcomes?
Well, but they're saying like ATM is an apical kinase.
Where do they say that?
In many, the first sentence of that paragraph. In many species, the choice of
Oh, by 3 apical kinases.
Yeah.
Oh, yeah. Okay.
So they must be apically localized.
Okay, weird. Sure. Sure. What does apical mean? For a whole creature. Okay, well, going on. We're moving on.
Yeah, I mean, this is a question that I just never understand what apical means. Ever.
The apical side of the lung epithelium is the side
Closest to the air side.
Yeah, closest to the air side or the
That's
lumen.
all I know.
Yeah, that's all I got.
Well, and I do feel like maybe part of it is semantics too, right? Like you can have an apical side of the cell, but you can have an apical side of the tissue.
And shouldn't those be the same side?
Yes, but they're different scales, right? Like you're like could be talking about
I will say,
like
you
a
should-
multi.
okay, yeah.
Me and you should keep our.
Okay. Anyway, I'm gonna move on.
You got to gesture with the left hand.
Okay, yes. Planarian flatworms have an abundant population of adult stem cells that actively divide to maintain tissue homeostasis and fuel regeneration. Exposing planarians to ionizing radiation introduce double-stranded breaks that induce apoptosis. High-dose radiation depletes all the stem cells within 2 days, while lower doses of radiation allow a few stem cells to persist. Although these cells slowly resume dividing, the animals still succumb, suggesting that irreversible damage has occurred. This eventual death suggests that despite survival at the cellular level, stem cells are unable to counterbalance DNA damage at sufficient rates to fuel long-term animal viability.
All right, now me. Even at radiation doses of 20 gray, which is the lower dose, the fact that some stem cells survive while others perish within the same animal suggests that factors such as cell cycle state play an important role in driving radiation resistance. Despite conservation of key cell cycle regulators and components of the DDR pathway, molecular mechanisms that govern planarian stem cell response to radiation are as yet unclear. I hate the word unclear in papers. I don't think there exists a paper that doesn't say it. Here
Do you prefer remains to be elucidated?
also hate that. I hate the whole like
Remains unknown.
A gap
Yeah.
in knowledge?
the whole thing sucks. It's all garbage. Here we capitalize on the radiation sensitivity of planarian stem cells to understand how these cells decide whether to repair DNA or undergo apoptosis. We find that after radiation exposure, ATM drives these cells to undergo apoptosis, and that depletion of ATM prevents radiation-induced stem cell death. Those stem cells that persist can proceed into the S and G2 phase of the cell cycle. These cells subsequently re-enter the cell cycle, likely through homologous recombination-mediated repair, and successfully fuel animal regeneration and repair. Together, our data uncovers a specific role for ATM in driving apoptosis after radiation, and highlights how the function of this conserved protein might have evolved in planarians. The findings of this study could, therefore, reveal mechanisms underlying stem cell rejuvenation that operate in highly regenerative animals.
Yeah.
That's the introduction. So I'll read, you're going to be the last person to read, Linnea. We'll read the first paragraph of the results up to the figures, and then we're switching away from the text. No longer reading the paper, just looking at figures, and we're just going to be confused. And that's just going to be a part of it.
Okay, before we move on,
Yes.
this is, I mean, I think I tried to ask Carrie this question in Woods Hole, but I still don't understand. If these creatures can do homologous recombination, why are they not genetically tractable? Like, why can't you put a plasmid in and have them uptake it? Any takers?
Megan?
No. The only thing I know is that there's something going on where they have a lot of pi RNAs.
Okay.
That like can any like
Can I ask you a
exogenous.
stupid question?
Well, I might give you a stupid answer, but okay.
What's a pi RNA?
It's like a small RNA that they're mostly expressed by the cell to silence transposable elements in the germline, to like silence like selfish genes. And they're like short RNAs that I think might get like transcribed and then like sliced up into like little components. You can stop me if I'm wrong.
I don't know.
We had a graduate student in our lab named Payam who invented a like optogenetics tool called PyTags. And I always thought the PyTags were named after himself. And now I'm wondering if it's related to piRNA.
Is pi RNA like PI?
Or is it PY?
RNA.
PI RNA.
Because the PyTags are PY, which is
Wow, that's very bold to like name your technique after yourself, but I like it. So in any case, I think
that makes it like really like any foreign DNA will get silenced.
I think so. I think these piRNAs can actually bind to and like chew up or like target these like foreign RNAs for degradation.
Okay.
And that's why it's been really hard to do any sort of transgenics in planaria, but I could be completely wrong.
Okay.
And there could be other mechanisms, but I do vaguely remember that kind of.
So like even if you got the DNA into the genome, it wouldn't, and it was expressing. You're saying that the RNA at the RNA level, it would get silenced.
I don't even know. This is a good question. I don't even know. I think maybe the limiting factor is once you get the DNA into the cell before it can do its thing to integrate into the genome, it gets silenced or chewed up.
Okay.
But I would imagine that would maybe be an issue too. Yeah. Like once it gets in the genome, maybe integrates into the genome, maybe it gets silenced.
Okay. Well, that was
but I feel
insightful.
Like maybe they did do transgenics in planaria more recently.
Carrie said they were still trying.
Okay.
Because I bugged her about it this summer.
Uh-huh.
And she said she has people in her lab working on it, but no one's quite as excited about it as she is. So.
Is there a planaria adeno-associated virus?
Probably.
Okay, sorry.
All right. Results.
So we're just reading up to figure 1A.
Yeah, I would just finish the paragraph, but yeah.
ATM is required for radiation. ATM.
Oh.
Is required for radiation-induced apoptosis of stem cells. To determine whether apical kinases...
What are apical kinases? Sorry, keep going.
They're kinases that localize to the apical side. I hope. Regulate stem cell responses to radiation in planarians. We used RNA interference, RNAi, to knock down key components of the DNA damage response, including orthologs of apical kinases.
What are apical kinases?
They're kinases localized. ATM, ATR, and DNA PK, all mutants. And the mRN complexes, mutants MRE11, RAD50, and NBS1. Which
those are just genes.
Those are genes. Why are they lowercase then?
So every model organism does it differently.
Okay, I'm
but
sorry.
Often,
I'm a noob.
Lowercase means a gene and uppercase means a protein.
Okay.
But yeast does it really weird.
Yeast does it.
Clammy does if it's lowercase, it's a mutant. If it's italicized, it's a gene, and if it's regular, it's a protein.
It could be something like that too. But I think based on the fact that they had the knockouts before, minus, I think
my
it's
theory
just
about why this is such like deeply hot garbage is that the biologists were jealous that the physicists had this notation that made things completely impossible to understand, and they decided that when they were doing genetics, they were gonna also get a notation that was completely impossible to understand.
I think that they think it's intuitive.
No, there's no way that a person thinks that this is intuitive.
It's in C. elegans, they do it really cool where like if you make a mutant, you get to like name that mutant and it gets to be like your signature plus like the number of the mutant. So like if MR hasn't been taken by another C. elegans lab, my like first mutant will be like MR-1 or something like that. And that's how they name all of their mutants.
Kill me.
Okay, these aren't mutants, these are genes. So, wow, this is still one sentence. I'm going to start the sentence over. Do people know where we are?
I would just move to the next sentence.
Okay, analysis of single-cell RNA sequencing databases shows that expression of these genes is enriched in stem cells as compared to differentiated cells. Figure EV1B. 1B, maybe extended volume.
Extended volume?
That's my guess. 2 days after...
That's your actual guess? You think that there's an extended volume portion of the paper?
Yeah, it must be.
What do you think EV stands for?
I definitely don't think that... Maybe extended video, but there's no way that it's a video.
It says figure. 2 days after exposure to 20 GY, we examined...
Gray. It's radiation.
20 gray, we examined stem cell distribution in abundance with in situ hybridization and QRT-PCR for the canonical stem cell marker PWE1.
PWE1.
Knockdown of ATM.
What is ATM?
Knockdown of ATM.
No, oh wait, I'm supposed to say it with apical kinases. Sorry, I'm just interrupting you. Keep going.
Of ATM.
Oh.
ATR and mRN complex components cause PWE1 cells to persist after radiation, while virtually no stem cells remained in control animals. Among these genes, ATM RNAi preserved the most stem cells as shown by PWE1 expression levels in QRT-PCR. Regardless of the RNAi trigger, based on these results, we conclude that the apical kinases ATM and ATR, but not DNA PK, regulate radiation-induced stem cell loss.
So sorry. I'm really giving myself away here because I have it in my proposal. What does PWE1 do?
So they always use it as a stem cell marker, but PWE is the piRNA pathway gene that like regulates parts of the piRNA pathway, but it just happens to be active in stem cells because stem cells need to express a lot of piRNAs to inhibit these transposable elements.
Okay.
But I mean, I actually think for our purposes, it's just a stem cell marker.
Just a stem cell marker.
Yeah, but it's a very highly conserved, like it's a stem cell marker in like many contexts.
Okay.
Okay, so if we go to the figure,
Figure 1.
figure 1A.
Everyone should shout figure 1 really loudly so that when I'm editing, it was so hard to figure out where to switch figures. This was the one took me time for the first one for editing. So just like figure 1.
Figure 1. Woo! Okay, so we have a worm, a whole worm, and we can see all of the stem cells in blue, is what Megan has just told us.
Yeah.
And then when they're irradiated for a random RNA, there's very little of that RNA left. Is that what this control is?
The next thing, they're using RNAi
Yeah.
to mess with different genes. So the first control is, I guess, they don't use RNAi or they use some nonsense RNAi.
Yeah.
The next one is they're going to knock down ATM.
Okay.
And so they see that the stem cells,
Recover.
that the PWE1 is recovering. And they say that the stem cells are recovering. The next one is they knock down ATR through RNAi. Is some recovery. And the last one is they knock down DNA PK through RNAi. And, oh yeah, I'm supposed to drink. No, I'm not supposed to drink because I'm talking. And they don't see the PB1 signal, so they say it didn't recover.
Okay. Sounds good. And I assume that B is just a quantification of this for more than one.
Sure. And then they have these MRE11, RAD50, NBS1. Why did we care about these things again?
There are other genes in the same DNA damage response pathways.
Yeah, so I think they're just showing that this like rescue is specific to ATM and ATR and not other DNA damage things.
Yeah.
So then they have these very physicsy looking plots. One DPI. What does DPI mean again?
Days post
irradiation. So 1 day post irradiation, they have a host blue versus host red. So like host blue is nuclei. I assume host red is also nuclei. What is this graph?
It's a cell sorting.
Yeah, this is clearly cell like FACS analysis.
So should we go back to the text?
No.
Or to the sub thing?
You think we should go to the thing? It just says FACS plot of host stained X1 cells from control and ATM RNAi animals.
Yeah, so maybe they're just showing that they're like alive.
What? Why?
Why are there two axes?
Yeah.
I don't know.
Both for host. So red and blue must be for different things.
They must be, one must mark.
Mark stem cells. And...
Yeah, well, it looks like they only took the ones that had high blue as stem cells. So I think they kept everything in the trapezoid, probably.
Mm-hmm.
But host is just cell nuclei, right? It's just like nuclear DNA stain.
But you must be able to separate out stem cells from non-stem cells based on the red.
Okay.
I don't know how.
Believe it. I believe it. Sure.
Are they the only ones? Does it show dividing cells? Are they the only ones that divide?
I can see a dividing cell on my host stains, and my host stains are blue. But also, there's a lot of different kinds of hosts. So,
Okay.
host, I don't know. Okay, then C is the quantification of that. Percent X1 cells. E is got a thing called annexin V.
Is it alive cells versus dead cells?
Percent of X1 cells.
Because
from
3,
control.
like...
You know what they do have?
Still, the higher percentage survive or are there or something.
Yeah.
Yeah.
They stained them with cell death marker annexin 5.
That's next thing. We haven't gotten there yet.
Oh, so maybe they're just showing that they sorted the stem cells.
I think they're quantifying, so they're not actually
Yeah.
like collecting. They're just
Okay.
quantifying everything in the box,
okay.
and they're saying that 3% of what's in the box is a stem cell.
Okay.
I will say we talked about this last time. Figure 1 is ATM drives stem cell apoptosis after radiation. It's a full sentence.
Oh, yeah.
Neither of us had heard of that rule before.
Well,
I feel like that's a common rule.
I think that might be a physics rule.
I feel that Martin does it too.
Okay, maybe it's, a stylistic.
Yeah.
E, you said annexin is a cell death marker.
That's what it says in the text.
Number
I don't know that
of cells.
A priori.
And then it's like a histogram. And then there's 10, 100, 1,000, 10,000. And they've shaded 1,000 and 10,000 in red to show.
You said it was a marker of cell death.
Less
cells die.
Less cells die when you inhibit ATM.
Yeah, which makes sense.
But what is the x-axis? The x-axis is how much of the cell death marker they put in?
No, how much of the cell death marker they measure.
Oh, okay. Okay.
So
I see.
if it's
I see.
above-
Yeah. So, like, I
1,000
assume they
die.
use the same amount of cells and then they.
Yeah. And then they quantify it in F because biologists feel like they need to quantify it.
Well, I think it's replicates.
Yeah.
In E, they don't show replicas.
Yeah, okay, yeah. All right, that's figure 1. Moving on to figure 2. RNA-seq analysis of stem cells following ATM. Not a sentence?
Not a sentence.
Not a sentence. A is a little diagram. Control or ATM RNAi. Then they hit it with radiation. They wait a day post injection of the RNAi. And then they isolate the stem cells. And then they plot a flat line.
Is it post-injection or post-irradiation?
Maybe it's post-irradiation. I remember during the course this summer getting that wrong every single day.
Yeah, well, it could be both.
post-irradiation.
In this case, it could be either, right?
Yeah, but it is post-irradiation, DPI. B is a volcano plot of differentially expressed transcripts. It really just looks like a line with a single dot that
Yeah,
says ATM.
That's the point. So they're showing that they have knocked down ATM and only ATM.
It's
so that's
very
actually very
specific.
important. Yeah, their RNAi did not influence anything except for ATM
Which is
in the control.
Okay.
So it's in part C that so only ATM was affected when they didn't irradiate. Right?
Okay.
Yeah, unirradiated.
Okay. And then they did irradiate.
and then
And a bunch of things
bunch of
changed.
things
bunch of
changed.
things changed.
Okay. And so then D is a heat map of unregulated transcripts used for RNAi in 2E. Looks like some genetic fold change stuff. So this is the genes that change when you do ATM,
When
Yeah.
when you do the ATM knockout.
you knock down ATM and do irradiation,
Yeah.
right?
Yes.
Yeah.
It's kind of weird that when you knock down, like you would expect that the ATM box on this would be like really red or something, right? Like you've knocked down ATM, shouldn't it be really
The
knocked
AT.
down?
Oh, I see what you're saying.
Yeah.
Well, I guess what they're showing is that ATM is upregulated.
But I mean, there's probably not that much of it there anyway. Is that right?
Well, it's fold change from pre-irradiated to post-irradiated.
Or probably fold change
Yeah, did
in
ATM
ATM
It's
RNAi
ATM
versus
the control.
But
And
then
so I would
what
have
is
expected
this
them to
control?
actually be the same.
That's like RNAi just with a scramble.
This
Is this saying that
showing-
ATM has
actually, what
been
is this showing?
upregulated when we knock down ATM?
Yeah, but maybe that's not so surprising. Like you're irradiating it, so ATM is going up, but because you've also provided RNAi, it's probably not going up as much as it should have.
It's confusing.
I'm confused, but we can continue on. And hopefully, it'll piece together.
Okay,
Do you
and
want to share
then
your confusion?
No,
Or are you just broadly confused?
no. I mean, I'm confused about this 2D plot, but
it's quite confusing. Okay.
So these genes, they're probably going to talk about later. The fact that they highlight them all here. I don't know what any of these do. A lot of them look like cancer pathway genes, right?
There's P53.
CHEK1, P53, RAD, I think is all.
And then cyclin must be a cell cycle thing, no?
yeah.
PW1, relative expression. Megan, you were going to explain it to us?
well, so 2E is just they do qPCR for PWE1 after using RNAi for all of these genes. So I think these are the same genes that are in the heat map. So I think they're just trying to see like PWE1, our stem cell marker, how is it responding when we knock down all of these genes? And I guess the takeaway would be that when you knock down ATM, PWE goes way up, which is consistent, I guess, with their in situs. And then also this other gene,
Chicken 2.
chicken2.
CHEK2, probably.
Is that like a checkpoint?
I think it's CHEK1, CHEK2.
Well, we could call it chicken 2.
But we can call it chicken2, that's fine. That also increases PWE expression when you knock that down.
And then they have nice little pictures of ATM knockdown and chicken 2 knockdown for PB expression.
Okay, so does this imply that ATM and CHIK2 are like somehow related?
I don't think so.
No.
Right, it's just if you happen to... Right, so they gave a
Are
mechanism.
they in the same
sort of-
pathway?
That's my question.
Oh,
Yeah.
what does chicken 2 do? I don't know. Well, we said it was a checkpoint thing, probably.
Yeah, so they might be in the same pathway.
But ATM is like DDR, isn't it?
Well, but
ATM
maybe
is
that could also
DNA
like
damage
affect.
response.
Yeah.
Chicken 2 is probably just like a normal, like, hey, you can't apoptose if you can't get past a certain checkpoint kind of thing.
Well, and to be fair, these are stem cells, so they're probably dividing a lot. So if you have like a cell cycle checkpoint gene that's going up, that probably works in a stem cell's favor. So maybe they're just saying, oh, it's increasing PWE and this thing that's
Yeah.
promoting cell division.
I think it's kind of just a by-catch that they just caught a thing by
Okay.
accident.
So this is where I do think I'm going to make a plug for the full sentences as figure caption. Is that what am I supposed to take away from this figure?
You're supposed to take away that they did RNA-seq analysis of the stem cells.
That is clearly not what I'm supposed to take away, right? Like I'm supposed to take away some piece of that this has taught us something.
That's fair.
And I want to know what it has taught me, and I still don't.
But yeah, that's fair. I do think kind of the cool, like, non-intuitive thing of this is like you're irradiating a worm, and so you're like giving it a bunch of like DNA damage. But then if you inhibit the thing that is responsible for repairing the damage, it actually fares better.
I don't like that we call them worms. Because worm like means a thing in my mind. It's like a tubular feature, and inside the tube is a smaller tube, and it like an earthworm.
Oh, you mean like a planaria?
No, because like two things. One, the planaria is like clear and flat, and second, the inner portion of it is like a weird branched tree, like it looks like a lung or something, right?
Would you prefer if I called it a flatworm?
I would.
Okay.
Do you think this is why you call the little caterpillar tokens and wingspan worms? Is because you think that they're little planaria?
No, planaria don't have antenna, do they? I don't think they have antenna.
No.
Okay, Figure 3. Again, I'm just going to do that real, like, figure 3, just to try to help myself when I have to put the pictures in. ATM knockdown via RNAi compromises the G1/S, cell cycle checkpoint, but not the G2/M cell cycle checkpoints after irradiation. It's a sentence.
That's
I added some words, but it's a sentence.
Yeah. I appreciate it.
Yeah. A, they hit the worm with 20 gray at time 0. Then they do an EDU pulse between 0 and 1 day. Presumably they're not doing a day-long EDU pulse. Or they do an EDU pulse between days 1 and 2. And then they fix an image. And they do this for the control and the ATM. In the control, they see that... So EDU, sorry. I actually know what this is, Lydia.
I know what this is.
Oh, you know what this is. All right, we all know what this is. So they see that there's no cell division going on for the control after they nuke it, and that if they do the ATM knockdown, there is division the first day. And there's no division the second day, which is kind of curious, actually. B is a quantification of that where they do EDU per millimeter squared. Seems unnecessary. But I guess this is...
No, this is, again, replicates. Seems good.
Okay, yeah. I keep missing the, replicate idea. C is the percent of G2/M cells, which they must get with another reporter. And this ain't showing replicates because there's 2 dots per graph.
I agree.
Oh, that's a replicate.
Sure. And then maybe I'll shut up and someone else will.
Okay, so what they see in the G2M is that the 1 day post irradiation ATM has more cells in this phase than the control, and same with 2 days. Which again supports the idea that they're still dividing, I think, because they're in different cell cycle phases.
Yeah.
D.
So D is pH3, which Megan actually recommended to me one day after I screwed up an experiment. Is EDU you got to pulse before you fix the sample.
Yeah.
But if you screwed up for some reason and you fixed the sample and you forgot what you were doing, pH3 is an immunostaining that will give you some cell division information.
Okay.
You can do it afterwards. So I have pH3 data for when I did a pretty big experiment and just completely forgot halfway through the experiment what I was actually trying to accomplish with that experiment. And I think I texted Megan that I screwed up, and she texted me, do you have pH3 in your lab or something like this?
Yeah, it'll just label dividing cells.
And so it's pretty similar information. They just do it for many more days. And I guess this is because the EDU pulse for a many day thing probably gets a little bit sketchy.
Yeah.
Yeah, and it's also like the EDU will incorporate into any dividing cell. So it will incorporate into any cell that's dividing at any point during your pulse. Whereas this pH3 will only label cells that are dividing in your fixed tissue at that time. So you probably just can't capture as many cells with a pH3 stain.
But it allows you to go much further after.
Yeah, and you can kind of just use it on any fixed tissue.
Yeah. And then E is H3P per millimeter. So this
is just the quantification.
The quantification.
Yeah. That the cells keep dividing if you've knocked down ATM. They don't really keep dividing if you didn't knock down ATM.
And they keep dividing if you've radiated them, but not if you haven't radiated them.
Oh, that's an interesting... No, it's the scale bar, right? The division rate, if you haven't irradiated them, stays at around 100, 200, and that's, I guess, supposed to be kind of just flat.
It's flat and not dependent on whether you have this ATM knockdown.
Yeah.
Okay.
So when they say ATM RNAi compromises G1S but not G2M, is the main data that in the 2 days post irradiation, we're not seeing cell division? Like, is that the main piece of evidence supporting that?
Yeah, I don't know, actually. Like, yeah, what actually here supports... Is this like a subtlety of the difference between how PH3 works and how EDU works?
I wouldn't think so. What do you think?
I don't know.
Yeah, well, they put that sentence in there, so that's quite nice, but I don't see what data supports that sentence.
Well, I think we can definitely say not G2M checkpoints. Well, I don't know.
They probably explained this very nicely in the text.
In the text, yeah.
That's not what we're
there's
doing.
lots of sentences.
I will say, the dark blue that people use for Dappy should be banned. It's completely invisible in everybody's images ever. Dappy's already kind of a shitty dye, and then they represent it using the most invisible color possible.
But it never tells you anything.
Yeah, but imagine how beautiful this image would be if they used two visible colors rather than a yellow and a dark blue that's completely invisible.
I'm sorry.
Hey!
Okay, so since
Look at
we
that,
understand,
figure 4, they use a normal blue.
and when since we understand the take home of figure 3, we're just not going to worry about the details.
I think we're just going for in this podcast, not worrying about the details. Like, we haven't read any of the text.
Yeah.
So, do you think in this blue of A, that's now Dappy as well?
No, it's Pee-wee.
Oh, sad. I hate this dark blue as Dappy. Okay. Stem cells repopulate and fuel long-term survival of ATM RNAi animals. So you have ATM knockout animals. Or ATM inhibited animals?
Yes.
Yes.
Again, a full sentence, so we know what to expect from this figure.
They have a Pee-wee 1 image, which just shows that there's more Pee-wee 1 when it makes me think of like
Tiny hockey players.
no, this is silly actor with like the red bow tie, like Pee-wee Herman.
Yeah, Pee Wee Herman.
Yeah. With like the weird, I don't think, can anyone do a Pee-wee Herman voice?
Absolutely
I've never
not.
seen it.
Yeah, they show an image. Looks like ATM knockdown keeps the Pee-wee cells happy. They quantify that because of replicates. That's why they quantify it.
Yeah. Very good. You're catching on.
Yeah. And then they do percent of Pee-wee 1 per animal area. They do it a second time.
So the number of stem cells goes way down and then recovers,
Yeah. This is what the...
but not as much in the control as the ATM knockdown.
Yeah. And it's interesting that it recovers it all in the control. Because I think
But you said that this 20 grays was kind of low.
This is the low. I think they described
Yeah.
it as low radiation.
Yeah.
Where do they say that it's 20 grays for ABC?
I just assumed because all of the last figure was 20 grays.
Okay.
In E, they do a huge dose.
In
but
E,
probably,
they do rads. I don't know what the conversion of rads to grays
Oh,
is.
those are, yeah.
I think it's a different thing.
Oh, really?
I do. I don't think a rad is a gray.
But I guess this
It's
is
so
lethal.
confusing.
Linnea is
Why?
doing the thing she did last time of trying to flip through the paper to find any meaning.
I am super annoyed that they would change units. They do
It's
change
probably
units.
a different machine or something.
Yeah.
So
yeah.
Yeah.
You think
'Cause
it's a
this one-
different machine?
Well, this one's the lethal dose, so maybe it's like way more intense. And so they used a different machine. I don't know.
All right, so they apply 2,000 rads 7 days after giving it the ATM RNAi. Then it looks like
7
they
days.
cut off its head.
Yeah.
Um,
well, because planaria can regenerate. That's
Sure.
You know that.
And they look for survival/regeneration. And they have a percent survival curve. In the control, whether or not it's been intact or decapitated, it dies 40 days after radiation.
Such a wild creature that you could.
What was the point of decapitating
head
it
off.
Yeah.
And
for
then
this curve?
in one of them, they all survive.
They all run their heads back.
As long as you knock out ATM, they all survive.
So we're assuming that 2,000 rads is greater than 20 grays.
Yes.
Yes.
You're assuming.
That's what I'm saying.
I'm not assuming that.
You're not.
That's
assuming.
definitely true, though.
What do you
Definitely.
mean? It's definitely true that 2,000 rads is greater
Because
than
when
20
they were
grays?
using 20 grays, they weren't just like straight up dying.
I don't
but here
even,
they've cut off a
I'm not
head.
even convinced.
But even the intact
I feel like
ones
we might
die.
be saying that 2,000 apples is definitely more than 20 oranges. That like
Okay, but that can still be true.
That is true.
I'm not even convinced that they have the same units.
But they
That's
use the
okay.
same symbol.
They do use the same radiation symbol.
Yeah. So.
So then they have a bunch of very cute pictures in which sometimes the worms have eyes.
I love the googly eyes.
Yeah.
ATM makes the eyes bigger. I don't think that's what I'm supposed to get from this picture, but that's definitely what I get.
I think
Grow
its
faster?
head is regenerating, so you can
Yeah.
see its
So I
eyes.
can see the eyes better.
Yeah.
I see. Okay. And look, they did Dappy, and it's not dark blue.
But they did something else and it's dark blue. So.
I think that's Dappy.
Oh.
I think by default, dark blue is always Dappy in biology,
Wait,
unless
no,
specifically said otherwise.
If you cut it in half, you get 2.
Yeah.
If you cut off what is the smallest amount that you can keep, and it will come back.
There's a paper about this, like this is like one of the first Planaria papers, and they do like 128ths or something like this. I think it's bullshit. I think it's just like a paper that's actually about like experimental error and not uniquely about like the Planaria. And then there are papers that show that like just like a couple cells will do it if you get the right stem cells.
It's about which cells you get.
Wait, is that what you're asking?
I'm just
What's
asking
the
what's
smallest?
the smallest, the smallest amount of the worm you could keep and get a worm to regenerate
I
from.
see.
I will say, you want to move that mic closer to Zach? No, push it way on it. Yeah, there we go. Okay. Even though it's super late and that's not going to help at all, and if anything, it's going
just
to make my
gonna move
life
away from
harder.
it. No, I'm just going to move away from it. It was a choice to get further away. So.
That seems pretty straightforward. Should we move on?
Okay, so what we've.
Oh,
yeah, what have we learned? I mean,
Stem cells repopulate and fuel long-term survival of the ATM knockdown animals. That they can survive big radiation.
okay, and this is
if you
because
get-
ATM knockdown somehow allows for more stem cell regeneration.
ATM knockdown means that their DNA damage response doesn't
Work.
work. And so the stem cells, well, screwed up and have all sorts of DNA damage, don't apoptose and can eventually restore the creature.
Okay.
I'm cheating. I
You
worked
remember
with
this.
Yeah.
But this isn't just like a giant cancer blob? Like,
I think maybe one way to view Planaria
Is that they
is
are
a giant cancer blob.
okay, so that's just their default state.
I wonder if you put a Planaria inside a mouse, is that the same thing as giving the mouse cancer?
No.
But it is an interesting question of like, okay, well, we've inhibited ATM and allowed this worm to survive this lethal dose of radiation. But at what cost?
Yeah, at what cost?
Like, is the DNA super messed up now? And like, this worm has survived and can regenerate?
Yeah, did you do any of
think-
these
yes.
behavioral assays? Or did they do?
I mean, I imagine 40 days is a long time for them.
I think, yeah,
I mean,
so
if
I
they're
think,
still alive 40 days later, they will continue to live, probably, right?
but
Yeah,
then,
so what we were doing over the summer was as they're recovering, right? Obviously, during the recovering period, their behaviors are inhibited because they're screwed up. But, as you get further and further into their recovery, are they behaving the same way as a normal worm, right? Are they physiologically the same worm? And one way that we were trying to do that was via behavior. I would personally do it via, like, checking their metabolism and stuff like this. But
Well, another thing is, like, has their genome accumulated a bunch of mutations that are going to lead to other defects?
Or can those be rooted out over time? That some of the stem cells haven't had much DNA damage, and the damaged ones do manage to repopulate, but over time they get rooted out because they're too damaged.
Like a question of like how resilient are they to the DNA damage?
Yeah.
there is something about drinking alcohol and being recorded that makes my nose super itchy. We talked about it last time, and it is just the same thing again this time.
You can itch your nose. I won't judge you.
Yeah, I've been itching my nose. Skip. Oh, there's the discussion. Figure 5. Figure 5.
Figure 5.
For myself. Animal survival after radiation relies on homologous recombination.
Okay, can
Okay.
we get a recap of what homologous recombination is from the biologists in the room?
Take it away, Zach.
So you have your DNA, 2 copies of it, and then when you have sequences that are the same, the same sequence on 2 copies, the 2 chromosomes you can combine across them, right? I mean, like am I describing this right?
Yeah. I
Okay.
think so,
Yeah.
right?
And so you can actually, if you say you have a mutation on one of your chromosomes and the other copy is good, if you have crossing over events, so basically when the sequence is the same, the DNA can kind of swap places and the chromosomes are kind of connected and then they become disconnected.
Okay.
You can fix an error on one of the chromosomes.
Okay, cool.
But basically, pieces of DNA are crossing chromosomes.
Yeah.
Sort of.
Yeah, or like you can use the other strand or maybe
fit,
even
yeah.
the other
To correct,
chromosome
yeah.
to correct
The other
a-
strand.
Or
yeah.
change.
Yes, yeah.
And
Which
how
is
is it known like that?
It's like how-
Is it going to go in the right direction?
No. It's
it
going to be
could go in
random.
either
Yeah,
way.
It will happen probably in both.
Yeah. Okay.
Well, then how does it repair anything? Isn't it just diffusing out
Well,
the
the
problem?
cell that gets the good copy survives, and the cell that gets the bad one dies.
Oh, okay.
I guess.
Okay.
Survivorship.
Cool. Okay. A through C, Kaplan-Meier survival curves of intact RNAi animals after radiation.
And they do a bunch of different
A bunch of different
RNAi
knockdowns.
things. What does this mean? ATM semicolon control, BRCA2 semicolon
Probably
ATM?
double RNAi.
Double RNAi?
That's my
Okay.
guess.
Okay. Yeah, so survival, not great when you mess with RAD51 or our control or anything other than you just inhibit ATM. Yeah, do they show anything else?
So there are things that make it worse than the control. That's purple and green, and their ATM is the only thing they tested that makes it better.
What happened to Chicken 5 or whatever?
Check 2.
Chicken.
I
Chicken too.
did read the text. It was an ATM effector protein.
Oh, okay. So then it's just kind of
Sorry, I
redundant.
cheated.
All right. There's no rule against reading the text, right? Like if people
There's
want
no cheating in Journal Pub.
I'm just not going to read the text ahead of time. Then they show D, which you got to tilt the paper for, which is Peewee in situ hybridization of ATM RNAi plus gene above. Oh, I see. Okay. So it's ATM plus a gene or control plus a gene. It doesn't really seem to matter what other gene you mess with all that much. ATM is the important thing. So how does this what does this have to do with homologous recombination? The text of the figure was that the animal survival after radiation relies on homologous recombination.
So when you get have the radiation, you'll get double-stranded breaks, and presumably you have to fix those to survive.
Okay,
Right.
but what does that have to do with the experiments here of just knockdowning a bunch of stuff?
So I think homologous recombination, it's not just like a passive event. Like the DNA is not going to do it itself. You need genes to facilitate, like the DNA checkpoint proteins facilitate this.
And so maybe some of the
There's DNA
things-
pathway repair or DNA repair pathways where there's a bunch of genes that
like
detect.
the Artemis ATM might be something that messes with homologous recombination.
Probably facilitates it.
Yeah, so RAD51 and BRCA2 are both proxy for homologous recombination.
Okay.
And then DNA PK, is that the other one? Oh, no, it's not.
No, DNA PK is the one that they determined is unimportant. It's like
that's
DDR.
not homologous and joining.
Yeah.
Okay.
So how do we make sense of, like, does that purple, that RAD51 ATM
But the
dies
rad-
really fast, but the ATM RAD51 Peewee still looks pretty present?
So the stem cells are still
I think you're supposed to be
surviving?
comparing the dotted line to the solid line in each case.
No, but she's talking
Yeah,
about the
Compared to this, compared to the control, these are dying faster.
the stem cells survive, but they rely on homologous recombination. So even though the stem cells are survived, they can't do homologous
They
recombination.
can't repair themselves.
Yeah, so
okay.
the animal still doesn't survive.
Okay.
Is I think the point of the
Okay.
figure. Okay.
Cool.
Last figure. Figure 6. A beautiful gradient
Schematics.
figure. Yeah.
Mechanisms underlying ATM-dependent rescue from radiation in Planarians.
Not a sentence.
Not a sentence. Boo.
Mechanism underlying ATM-dependent rescue from radiation in Planarian.
Here's the pathway we needed to be able to see before.
Oh, yeah, this
Uh-huh.
is nice.
Okay, so ATM goes to chicken 2, goes to P53, goes to the G. So why wasn't this at the beginning of the paper?
Yeah, Zach just said that.
Cool.
Okay, and then there's some of these things are missing from Planarians.
Senescence.
So they're
Senescence
missing
and the
like,
check to P53 arrows.
So what are we? Schematic of the dynamics of stem cell abundance after radiation. X-axis represents the time after radiation. Y-axis represents the abundance of stem cells. Bottom boxes indicate underlying molecular mechanisms.
Okay, so when we radiate, in the first wave, which generally involves apoptosis, stem cell abundance goes down normally. But if you inhibit ATM, it doesn't go down quite as much. Then in the second wave, where there's also apoptosis, things level out. And then in step 2, where the stem cells repopulate, the ATM RNAi is able to functionally recover, which means the stem cells can then repopulate the animal. But in control, they're not able to do that.
Okay, good summary.
Yeah, so, but this relies on that there's a very small difference in the second wave apoptosis floor.
Yeah, why did we? Were
Which-
there ever any like figures about a second wave apoptosis?
Yeah, well, I think this
So, I
was
think
the
there
first figure.
was.
Oh, no. When was this?
Because we
This
kinda
was
talked
the
about this.
figure 3, where it's 1 day
Yeah, probably they
versus
had
2
the,
days.
okay.
Yeah.
and then they specifically used.
So it's just like small differences in the number of surviving stem cells make big differences in your ability to recover.
Yeah. So, in conclusion, our study identifies a new role for ATM in planarians influenced by the unique biology of these animals. While its function in regulating checkpoint progressions and apoptosis is shared with other systems, its role in promoting stem cells exhaustion seems to have diverged. This difference may be due to the absence of significant portions of the protein or from evolutionary divergence of the pathway. There's a whole thing that we didn't get because we didn't read the actual paper. The evasion of ATM-induced apoptosis opens the door to elucidating the role of checkpoint proteins and DNA repair pathways in these fascinating stem cells.
Cool.
Cool.
I mean, I'm excited.
Wait, where?
I don't think
what
I.
was that?
I don't think
That
I
was
grasped.
the very last paragraph of the very last page that I
Oh,
printed.
okay.
I don't think I grasped everything in the paper, but I am still excited about planarians.
All right, Megan, what do you think it means when they say figure EV2A? What do you think EV stands for?
I thought Linnea's So what did you extended
Volume.
volume? That sounds right.
You think volume is correct?
Okay, this is EMBO.
Extra, extended.
Extended sounds right.
What would V be?
What would V be besides volume?
It wasn't a video.
Verbosity.
That sounds less likely than volume.
I agree, but it is embo.
I'm just saying maybe
we
that's
have the
what they
methods?
call their supplementary.
Just the first tiny bit. Not really.
I'm assuming on the page, like usually you can like download supplementary
Yeah, I'm
material,
sure I
so there's
could go
probably
check.
a button
won't,
that
but
says like download EV1.
If we check, we'll put
It's
it
a
in the.
Pokemon. Eevee.
Is a Pokemon?
Yeah.
I mean, maybe that's what they're referring to here.
Yeah. Closing thoughts.
Yeah, what did people think was beautiful about the paper?
I mean, I do think like it's a very unexpected result, and they, resolved the mechanism, I thought, pretty extensively. Given the tools that they had, like this idea that, if we actually inhibit this thing that we know is really important for DNA damage repair, it actually improves survival in this radiation model, which is like kind of it's like a very like surprising result to start with. And they're just like, they're clearly kind of just scratching the surface of like how this is working. And presumably, based on Bez's account, they're still trying to kind of figure out what the long-term consequences of this are.
I just love the model that like it's an immortal model, and then they figured out how to make it unimmortal by nuking it. So now you've got a model that is immortal, and you can unimmortalize it. And now you have a trick where you can make it get unimmortalized back to being immortal. So you have this great mortality switch that just like makes studying the concept of aging, which I put in quotes here because this thing is like immortal, really fun. That this is like the funnest aging model to me because of this wacky switch that you can do. And now they have like a recovery to this wacky switch. That's what I love.
That's cool.
And if you could have one experiment added to the paper, what experiment would you add?
You're really doubling down on these classical journal club questions.
No, I think that's
Yeah, it's great. It's great. If you could add one experiment to the paper, what would you add?
I would I want to see long-term, like they go out to 40 days, but I think maybe I would want to go over several generations of, what do they call it? Like splitting?
Yeah,
breaks, or maybe, or like several iterations of like we cut it in half and then it made a new one, just to make sure that it wasn't this thing that we were talking about where like it survived, but at what cost? But it has all these mutations. Like I'd actually want to know versus a control in a long-term setting where we're going over multiple generations of the like splitting and regenerating, if it's able to keep up with worms that never experienced radiation in the first place.
Yeah, so like what is ATM doing? Like why is it, if it's not helping,
right?
Why is it there?
I would like it's essentially the same thing. It's a little bit of dirt. I would like a physiological readout that's not % survival. I would like the thing that in my mind is like oxygen consumption rate over time.
Yeah,
but
they're all in the lab, so like they're getting all the nutrients.
Yeah, any physiological thing that moves around with kind of like how healthy it is.
Can you put planaria in these, what are they called? Seahorses?
Yeah, so I emailed Carrie to see if she would ship me some, because I'm pretty sure the answer is if you do it right, yes.
Okay.
That the standard seahorses will just squash them into pancakes. More pancakes, thinner pancakes. But the one that we have, I think you can do this on.
Oh, that'd be super cool.
Yeah, I really want to do that.
Yeah.
Okay, I have another experiment. Sorry, I'm being selfish. So, like, they show in the RNAi, like 2 days post irradiation, all the stem cells are survived, but then a lot of them die, but then it's able to recover. And so I'd like to see, like, 2 days post infection,
irradiation.
whole genome sequencing on the
That's right.
on
Yeah.
the stem cells, right?
Well, I was thinking whole genome sequencing after 40 days or whatever.
Long reads.
Why are they different?
Yeah, or like your suggestion of like, oh, is it able to sort out, like, to selectively only use the stem cells that don't have a ton of DNA damage? Because then we could actually verify that it's like doing that it's like only keeping stem cells that have managed to repair. Oh
We've
no!
lost the thing. Okay. So at this point, we should probably end.
Does that have sound?
That'll have sounds. The last
okay.
bit will be bad sound. Why am I still? I'm still going like this. All right, everybody say bye.
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 Good